Hey, welcome back. We are now finishing up our last section of this chapter. We had finished up with, we did lipids, we did carbs, we did proteins, and now we're moving on to the final category, nucleic acids, and the pieces that make up the nucleic acids. So our fourth organic molecule, like carbs and proteins, nucleic acids are also chains of repeating pieces, the same kind of the same sort of unit repeated again and again. Those repeating units are called nucleotides. So we've got a couple of different major kinds of nucleic acids. What they have in common is the nucleic acid part and the names deoxyribose, the D for DNA, and the R of RNA is ribonucleic acid. These are the genetic information of cells and viruses. In addition to just being repositories of information, RNA can also do a few jobs. Like it can act as an enzyme, it can help bind the amino acids to each other, helping to form the polypeptides. And what I'm going to remind you of is, there's messenger RNA, mRNA. That's the one that includes the instructions or which amino acids you should put together. But then we also have tRNA, the transfer RNA. That is the one that's reading the RNA on one side and bringing the correct amino acid on the other side. And rRNA, the ribosomal RNA, makes up the ribosome. Okay, so it's acting like an enzyme. All right, let's see. Nucleic acids, we said they're made up of these nucleotides, these individual units. Sometimes you'll hear a structure called a nucleoside. It's not wildly different from a nucleotide, but let's go over the nucleotides first. So like amino acids are the building blocks for proteins, so nucleotides are the building blocks for nucleic acids. Like amino acids, they're also made of three parts. There's a phosphate group, there's a sugar, and there's a nitrogenous base. So phosphate groups, always the same. The sugars, there's a 5-carbon sugar, pentose sugar. And then depending on one of the side groups, we either call it a deoxyribose or a ribose, and I'll show you the difference. And then finally, the nitrogenous base, you know that in DNA, there's 4 different ones, A's, T's, G's, and C's. And in RNA, there's 4 of them, A's, U's, G's, and C's. And I'll show you that on a figure. And then the nucleosides, it's just imagine the nucleotide, phosphate group, pentose sugar, and a nitrogenous base, except we just have the sugar and the nitrogenous base. This phosphate group is missing. That's what a nucleoside is. So I'll show you. All right, so here's the stripped down, like this is the generic nucleotide. There's the phosphate group, always the same. The pentose sugar, a 5-sided sugar. And then the nitrogenous base, this is the piece that varies. Kind of like those R groups, the side chains on the amino acids. There's different versions, but there's not 21 of these. There's only 5 different ones. Okay, now let's take a closer look at the sugar. What they have in common is that they're 5-sided. Look at this one functional group. On this molecule, there is a hydrogen hanging off by itself from this carbon. And on the other sugar, there's, instead of a hydrogen, there's a hydroxyl group. There's an extra oxygen on here. I kid you not. That's the difference between ribose and then without the oxygen, deoxy, without oxygen, ribose. That's the big difference. Okay, now let's have a look at the nitrogenous bases. We said in DNA, there's 4 A's, adenine. So A's, T's, G's, and C's. Adenine, thymine, those can bind to each other. They can make 2 connections. And guanine and cytosine, those can bind to each other. They're attracted to each other, but these only have 2 connections between them. I'll show you on another diagram. And then finally, so three of these are the same. Adenine, guanine, and cytosine are the same in DNA and RNA. The one difference is this. In DNA, we have thymine. And look at this. This is quite similar in structure. Let's see. I'm trying to quick find the, oh, there it is. It's this one group. Here's a methyl group, and here's just a hydrogen. That's the big difference between thymine and uracil. So uracil is going to bind to adenine. A and U go together in RNA. Okay, next slide. Let's see. Nucleic acid structure, it's a long chain of these nucleotides, just like proteins were long chains of amino acids. There's a covalent bond between the phosphate of one and the sugar of the next one. Just like with amino acids, the carboxyl group and the amino group held hands. Here it's sugar, phosphate, sugar, phosphate, sugar, phosphate. Okay. Like we said, cytosine and guanine are attracted to each other. They form three bonds. Adenine and thymine are attracted to each other. If you need a way to remember that, as a student, I could not remember which one went with which. The curvy letters go together. C and G go together, and A and T go together. And then the tricky bit about this is you have to remember that T and U, thymine and uracil, are kind of substitutes for each other. The way I remember it is in the alphabet, T and U are right next to each other. So those are kind of buddies. So they replace each other. Let's see. The DNA molecule, deoxyribonucleic acid, that tends to be two strands wrapped around each other. We refer to it as a double helix, double-stranded. The two strands are complementary. That means that if on one side I have an A, on the other side I have a T. If on this side I have a C, then on the other side I have a G. They're partnered up like that. And we say the two strands are anti-parallel. Let me remind you, parallel would look like this. As a student, I thought, well, anti-parallel must be that. No, anti-parallel means it runs the other way. So you read them in opposite directions. All right, so here's a close-up of just the one strand, alternating phosphate group sugar, phosphate group sugar, phosphate group sugar. And then there's the nitrogenous base kind of hanging off of the sugar. And that's how it can have another side that, you know, if you think of them as flags kind of facing in towards each other. Wherever you have an A on one side, you'd have a T on the other side. And that's a so-called complementary structure of the DNA. And that's the giant secret of how we maintain genetic information, how we make copies of the genetic information and can pass it from one cell to the next cell. All right, so what do we use nucleic acids for? As we've mentioned, DNA is the genetic material of every single last living cellular organism. Bacteria, archaea, everything in the eukaryote, I mean, all of those cellular organisms, every last one of them. On top of that, a lot of viruses also use DNA as their genetic information. The thing that's a little bit funny is that there are some viruses that don't have DNA. They use RNA as their genetic information instead. That's a little bit funny and interesting, but none of the cells, no kind of cell can use RNA as strictly as its genetic information. They all have to have DNA in the cells. We do also use RNA, and we use RNA to carry information. So there's messenger RNA that provides the instructions for putting amino acids together in the right order, the right primary structure, to make the correct polypeptide chain so that it can fold and become a functional protein. This DNA it controls the synthesis of all of the molecules of the organism, because it controls the synthesis of the proteins, and then some of those proteins are enzymes that control the synthesis of other molecules. a comparison of DNA and RNA. They're both nucleic acids, which is why they have the Na in common. One difference is the details of the sugar. Remember that the ribose is that five-sided sugar. Deoxyribose is a five-sided sugar missing an oxygen. Let me think about how much I want to spend time on this. Now I'll back up for a second. All right, so we talked about the A's and the T's and the G's and the C's. Okay, what I glossed over here is, if you notice, these nitrogenous bases are double ringed. They're a little bit extra wide. And then these in this column are single ring. They're a little bit narrower. And that's what we mean the complementary. So they're almost like when they bind to each other across the way, you can kind of think of it as being like three rings wide. Because A would bind with either T or with U. If we're talking about RNA binding to the DNA, or we have guanine binding to cytosine. Okay, so the two ring structures are referred to as purines. The one ring structures are referred to as pyrimidines. The way I remember it is silly. Purines is the smaller word, but chemically it's the bigger structure. And pyrimidines is the bigger word, but it's the smaller structure. Okay, let's move forward back to here. So the purines, we have A's and G's. They have that exactly in common, DNA and RNA. And then the pyrimidines, that's where there's a little bit difference. They both use cytosine, but the other one is either a uracil in RNA or a thymine in DNA. DNA tends to be double-stranded, and in RNA tends to be single-stranded. The exception to this is in retroviruses. There should be a T there. They carry, well, I'm not going to go into that in too much detail. Okay, and then function, the genetic information of all the cells and the DNA viruses, and the function of RNA in our cells is protein synthesis, and in some viruses, it's also the genetic information. Okay, and finally, last concept. This is also under the category of nucleic acids, but it's actually like one of these units. We talk about this all the time. ATP, it's that energy molecule. This is the cash of the cell. This is what we use to pay for things all the time, all over the cells. We use it, bacterial cells use it. It's just the currency of life. And that's what they mean by short-term energy supply for cells. It's used super fast and recycled super fast. How to think about it. So what ATP stands for is adenosine triphosphate. When it has all three of the phosphate groups on, it is at its highest energy form. And then in some ways, it can transfer the energy when we take off that third phosphate group. We go from ATP down to ADP. We release energy. And so that ADP is not done. We need to recycle that thing back into ATP using energy from another source, and then we can reuse the ATP again and again. Okay, so a diagram of it. Remember we said that nucleotides have a sugar, a phosphate group, and a nitrogenous base. So here's our sugar, ribose. Our phosphate group, just pay attention to one of these for a second, and then the nitrogenous base, adenine. That's the A of the ATP. Okay. If you just have the adenine and the sugar, that's where sometimes you'll hear this term nucleoside. It's not like a wildly different structure. It's just looking at the nitrogenous base and the sugar without the phosphate groups. All right, fine. If there's one phosphate group on, it's called adenosine monophosphate. 2, adenosine diphosphate, and 3, adenosine triphosphate. And where we usually will talk about it is kind of as it wobbles back and forth between being ATP, adenosine triphosphate, and ADP. So This is the high energy form when it's got all three of the phosphate groups on. And then the slightly lower energy form is when it's taken off one of these phosphate groups, released its energy when it takes off the third one. Okay, and then we recycle that thing back into ATP using oxidative phosphorylation or substrate level phosphorylation. It's a fancy way of saying phosphorylation is a fancy way of saying I'm putting that phosphate group back on there. Crank up its energy level. Okay, we'll get to that in the next chapter. And that is it for ATP. And then finally, I want to leave you with this thought. Thinking about the cell membranes of lots of different organisms, we all use phospholipid bilayer. So we're moving back to something that we talked about earlier in this chapter. But if you look at the cell membranes of microbes living in the Arctic, usually we see a lot of unsaturated fatty acid tails on those phospholipids. When at the same time, if we're looking at the membranes of microbes living in, for example, hot springs, which are very warm, you see more saturated fats in those. What function do you suppose that might play? I'll let you think about that, and we'll come back to it later. All right. Hope you have a great week, and I will talk to you then. Bye.